Okay, good afternoon. My name is Suxia. That I'm associate professor in the Department of Electrical and Computer Engineering at Prairie A&M University. So today I'd like to share with you my research on face identification over historical motion picture film. So this is a project that I'm collaborating with Moving Image Research Collections, MIRC, at the University of South Carolina through my XSEDE Campus Champion Fellowship. This project started September 2020, and Dr. Greg Welsbacher is the PI. And then we collaborated with Dr. Song Wang and Ms. Jung Zhou from Computer Vision Lab at the University of South Carolina. First, let me use this opportunity to show my appreciation of this fellowship program. My coordinator, Dr. Alan Greg, Dr. Jay Alamanda, and Dr. Robert Sinkrowitz, and all my collaborators from the University of South Carolina. So it is worth to mention that Prairie A&M University is an HBCU. Although my department has PhD program, but Prairie A&M University is still a majority teaching institution. And so we have limited resources for research. This fellowship opportunity greatly enhanced my research capability in computer vision, and I wish that more researchers can benefit from this like me. So now my presentation that has those outlines with those introduction, and then following by the methodology of our choices. Also experiments and analysis at last will be our conclusion. So these projects deals with unique data sets from United States Marine Corps Film Repository, who is a partner with MIRC at the University of South Carolina. So they store thousands rolls of World War I, World War II, up to the 60s and 70s historical films. So a big hurdle is to scan and store them, to categorize them with unique features or person of interest will help to maintain the database. So our research is to automatically locate the key personnel in the films, label them for future research and education. So we started with President Johnson, since he has enough available photos and videos to build the training set. So our project challenge are obvious, since most of those current face identification approach are based on the modern digital camera with high resolution. Our videos are mostly with low quality fog and blurry features. So if we apply the current popular method, there are misrecognition as shown in these slides. So you can see that those boxes in red that marks the confidence ratio of identify a person, but those examples show they're either identify or couldn't identify the correct person. So the goal of this study is to build a neural network with deep learning techniques for historical video-based face recognition. So there are three folds of our methodology. First, based on multitask cascaded CNN, which is called MTCNN, proposed by Zhou and Cheng in 2019. We propose a trunk branch concatenated MTCNN. We call that TB-MTCNN to enhance face detection performance, followed by RNAseq as recognition tool. Second, we incorporate object tracking algorithm into our system by adding deep data association, which is also called deep sort. Third, we apply image restoration scheme to reduce the effect of noises and blurriness. These slides show the system diagram. Specifically, the TB-MTCN model consists of 3 subnets. So let me go to the next slide first. So this is the majority of the contribution of our research. So those three subsets, we call that PNet, RNet, and ONet. PNet is for proposal network, which produce all candidates boxes with this probability of a phase. RNet is refinement network, which refine the boxes from the PNet and drop the candidate boxes with those low probabilities. And then the ONet is the output network, which produce a final bounding box. So the three stages of those deep convolution networks will predict phase and landmarks location in those subset networks using those cores to find methods. Extra branches are added to the convolution. As you can see that the basic branches that are marked with blue, but on top of that, we actually also add those branches for those convolution using those 5x5, 7x7 filters that diversify those feature selection, which will improve the performance of only those 3x3 size filter provided by the original MTCNN. So this trunk network learns facial features from those blur historical film images, while those branches network learns the facial features from the low-level feature map generated from the trunk network. Richer facial features can be extracted with those multi-scale respective fields by assembling various size of those convolution filters into the network. The lower level layers are shared by the trunk and the branch networks to reduce the computational cost. So to perform these comprehensive phase features, and then the output features map can be fused by concatenation. So this is our proposed TB MTCNN structure. I come back to the whole system diagram. So another insight of our method is that the image phase detection algorithm may fail in some of those frames. We can recover such missed phases by tracking the same object in the continuous frames. Specifically that the TB MTCNN model is applied as a phase detector to extract the facial features and to predict the phase bounding box. Simple online and real-time tracking with deep data association, also called as deep sort, as an object tracking method, which will integrating those common filter and intersection over union, IOU distance algorithm, also combined with those concurrent data association algorithm. This algorithm is applied to phase tracking and phase bounding box updating. Subsequently, that those updated detected phase bounding box will be transferred to the ResNet for recognition, and also classifier of particular phase. So finally, we get the historical figure phase recognition. In addition, that you can see that at the bottom, this dashed block that is actually shown in for these deep sort stages. Also, at last, that we apply image restoration method. We actually pick one of those very mature methods, which is called bringing old photo back to life. So this is proposed by Van and Zhang in CVPR 2020, which is specifically designed for phase restoration from old photos. At the beginning, that I said that this process that can be divided into 3 stages. For this last stage, at this point that we adopt this phase restoration method, but we are still working on how to use a more light-weighted image super resolution method to replace the last part. Okay, so for our experiment, that those results show that our proposed TB MTCNN outperformed original MTCNN and other state-of-the-art method. we choose 2 stages CNN. As you can see from the comparison of the result, 2-stage CNN, FaceNet light fast phase detector, which is LFFD, and also CMS RCN, along with original MTCN to be the other 5 comparison methods. So the last row is our proposed method. To be convincing that besides our unique historical film, we also add other popular data sets. Like this one is actually from the wider phase data set. And then the next slide, this is from Suxia web phase. And then for these slides, it actually includes our unique data set. You can see that we actually choose those metrics, including precision, recall, and F1 score to finish this comparison. So those numbers that gradually shows this improvement of our choices. Also, as we mentioned, that adding those deep sort method can further improve the tracking and for the face recognition. So this slide shows that by adding this for those object tracking, the percentage of the correct identification can be improved. So as from these slides, you can see that those undetected faces can be detected through object tracking. So next slide, it shows the visual improvement after image restoration. So finally, the recall and the precision of all the improvement after TB, MTCN are shown in these slides. So the first row, you can see that this is obtained from the still image by single image face identification. We can reach around the precision of 68%. So you can see that the recall rate is 68%, which means that the total of 300 frames with President Johnson's faces in the video, that 68% of them can be successfully identified. So with the step-by-step improvement, we finally can reach nearly 88% of those recall rate. So finally is our conclusion. To conclude from this research that a neural network model has been built to accomplish those face detection and recognition. Our contribution that include the face detection model was optimized by concatenating trunk and branch from the model MTCNN. And then we can extract the face feature sufficiently. Second, that to compensate the face missing detection among different frames that the object tracking algorithm was integrated into the face detection neural network. Moreover, that the state-of-art image restoration method was used to reduce the effect of noises and blurriness. So among those conclusions that from those research that we observed those effective ways to deal with those motion pictures in the 60s and 70s of this range. So in our future work that we are going to revise those image restoration methods into our framework to have a lightweight super resolution approach, especially right now that we research on that if with those super resolution method that if no ground truth is provided, so how to effectively finish those super resolutions. So that is our last stage. And here I list several of those key references. So I numbered them, that's because we co-authored a paper. So those are the numbers. As you can see that the tables is actually cited by the numbers from the pending paper that we submitted together, okay? And then thank you for listening to my presentation. Any questions, or you can contact me that I put my email on this slide. All right, yes, thank you.
Thank you, Suxia. We have time for one quick question. Do we have any questions?
Okay, so I can stop share my screen, so you can.
Suxia, let me ask one quick question, if nobody has any. With the image restoration, so do you use the knowledge? Say, for example, you want to improve the quality of the image of Lyndon Johnson's face. Do you use the knowledge that is Lyndon Johnson to do the improvement?
Okay. Good question. Yes. So as I mentioned, that this ongoing part is what you just mentioned, because there are strategies. For example, that if majority of them of the current image data set, that if we can easily build the ground truth image. So majority of the work is done from that. Our work is actually, we don't have the ground truth. Most of them, because from the videos, we actually will going to deal with 200 Navy generals. So those generals, they pretty much don't have that many face or clear faces to build the pool. But for President Johnson, he is unique. We can currently download a lot of just very clear images from him. So currently that we are using his image with his information to build this database to detect those, and then try to use this as the similar model for the degradation of those years apply on other generals. Yes, so yes, thank you for asking these questions.
Great, thank you. Okay, so we're gonna go on to, I'm sorry, we're just a little over, but I want our remaining presenters, to use your full slot. So our next speaker is Kurt Showmaker, University of Mississippi Medical Center. So Kurt, go ahead and share whenever you're ready.
Okay. Can we see my slides?
Yes, we can. Take it away.
Thank you. Today I'll be talking to you about my fellows project, the scanner, the single cell web annotator and web visualization app. This was South Carolina's Gushani's PI, and Chuhan was my ECSS mentor. The project description for this project is that single cells are the building blocks of tissue, organs, and organisms, and that the cells can shift among different biological states. And the analysis of these types of data is what this tool is designed to do. And this single cell RNAseq is becoming very popular, so we developed a low weight, low friction web app. And when I started this project, they already had a bioRxiv publication, which is here on the left, and it describes how scanner is used by a bioinformatician, in which the bioinformatician runs a single cell RNAseq analysis with the R package Seurat, and then uploads it to scanner, and then the biologist is able to get into scanner and then sort the data. So it is again an R-Shiny app that is deployed. Today's talk, I will talk about the biology of single cell RNA sequencing, the scanner app, and a quick tutorial. So the main premise of single cell RNA-seq is that cells differentiate. So here on the left, we have a club cell in the lung that differentiates into either a goblet or a ciliated cell. As well as on the right, we have a stem cell that can differentiate into a blood cell or a monocyte, and then further differentiate into different macrophages. So traditionally, when we did RNA-seq or gene expression analysis, we would take a piece of tissue and homogenize it from, a treatment group, like a healthy lung versus a diseased lung from a smoker. And then we would isolate the RNA, we would make libraries and then sequence. And by traditionally, I mean the last 10 or so years. And when we get done with the sequencing, we have the raw sequence output that is about 250 gigabytes worth of data. And in this data, we have 8 samples if we did 4 reps for each treatment group, and then 34,000 genes. And we would look at this data in volcano plots, PCA plots, and heat maps, and then call the differentially expressed genes. And then each sample would have its own barcode. So to get at a little bit more fine group, we would do different techniques, such as taking the tissue, isolating single cells, and then once we have a single cell suspension, we can either then go pick out single cells and put them into a 96-well plate like this, and then make individual libraries, or we can actually, type the cells by using antibody labeling with a flow cytometer. And then we would take the cells and we can either do bulk RNA-seq again, or we can isolate single cells from there. And that would give us a refinement to say, this is this cell type because it has this marker on the surface. However, we went through a technological revolution with this microfluidics, where we now can take the tissue, isolate the single cell suspension, and then run it through a machine like 10x Genomics Chromium, in which that we can bind each individual cell or nuclei with an individual bead that contains a specific barcode. And then we can demultiplex the barcodes after sequencing, and we can look at the data in whole. So every cell has a unique barcode, and then we're able to visualize the data with UMaps or t-SNE plots, like the UMAP plot shown here. And interesting, here we have 4 samples, 2 of which are control and 2 disease states. And on the right, you can see here that the group number 7, the cluster 7, is not present in the control, but does show up in the disease. And that is the type of data that we really like to look at inside the single cell RNA-seq analysis. Interesting that data dimensions have changed a little bit. So now if you have 4 samples, you have 30,000 genes or so, and times the number of cells per you isolate. So if you isolated 18,000 cells per sample, that would be a lot of dimensions. Usually it's around 10,000. Okay, so back to the scanner app. The scanner app, again, runs on an RRDS file. So you run Seurat just like you normally would, and you export an RRDS file. You run a little bit of a scanner-specific script to make sure the columns are in the right spots inside the RDS file from scanner, the Seurat object. And this Seurat object contains the sample, the cell metadata, the sample origins, the cell labels, what types of cells that we've predicted they are, this gene expressions, and the marker analysis. The RDS file is about 10 megabytes in size compared to the raw data, which is 250 gigabytes in size per run. So again, it saves a lot of space doing that. And the scanner app also contains the gene ontology, the KEGG pathway analysis, and reactome pathway analysis. So this is the system architecture of the scanner app, is that the users log on to the RShiny app through the internet, and it is hosted on Jetstream, running on a Docker image with Firebase authentication and Firebase real-time data management. Scanner is free, as in free beer. So it's free for use, but Scanner is not an open source project at the moment, and the source code is not available. So that kind of limits what you can do with this platform. Some of the current challenges we've faced and solutions. Well, the challenges are that Scanner was highly dependent on the Seurat object. So it had to be a specific version of Seurat. Otherwise, Scanner did not know how to handle small changes. It only works with human data at the moment, only works with one organism at a time, and it lacks support for some cell analysis, some single cell analysis like cell population proportion changes and cellular trajectory analysis. So to fix some of these problems, I built or at least the Seurat dependency problem, I built a singularity container that pulls the singularity image from the Seurat image from Docker Hub, and then installs the scripts needed to run the preload scanner scripts. And then we added support we looked into adding support for annotation, DBI, and biomarker to easily add organisms that has not been implemented. And we were looking into, custom support for annotations that are not currently in there, and to add a broader range for such as like agriculture, because right now it's only human data, and stuff like agriculture just doesn't work very well with it. So into the tutorial, This is an example set of 6 smokers and 6 never smokers from 2019. This is preloaded on the scanner app. So if you go to the scanner application website, you can go ahead and look at this data. It has 6 cell types, including a 7th cell type that seems to be unique to smokers, the basal smoking group. So again, here's the cells and where they're located on the plot. And this is just a Shiny app. So like all Shiny functions, you can change the data up by split it by age or sex. And then here we split by smoking, and we see some interesting results here where we can have this group that is only present, the basal cell that is only present in the smoker is not present in the never smoker. And we can see a big differentiation between the club sequences and the goblet cells. So we can also look at specific gene expression patterns. So here we're looking at the carcinoembryonic antigen-related cell adhesion molecule number 5, which is a known, has known association with tumors. And as you could suspect that if you split it by smoking, non-smoking, you see more of the marker present in the smokers. You can also display the data as violin plots, and this is the marker analysis, showing which genes are most associated with specific clusters. You can have your traditional heat maps per cluster, and here are the citations. And a disclaimer that scanner is not intended to treat, diagnose, or prevent disease. And I'd like to thank the ECS mentor Shuhan, the project PI Gusani, and especially the fellowship leader program Jay and Bob, as well as my fellow fellows. And that is all I have here.
Great. Thank you, Kurt. Excellent presentation. Do we have any questions? Right. Well, Kurt, I don't have a question, but just one comment. I want to say, I've been, exposed maybe tangentially to single cell RNA-seq for years. And that was the first time that I really understood it. So thank you for providing that excellent background as part of your talk.
Thank you, Bob.
All right, do we have any other questions, any more comments? Again, thank you, Kurt. We're gonna go to our next speaker, so Zhiyong Zhang at Stanford University. So Zhiyong, whenever you're ready, go ahead, share your screen and you can start presenting.
Okay, good morning, everybody. So can you hear me okay?
Yep.
Okay, so my name is Zhang, and I'm from Stanford University and work at the Stanford Research Computing Center. And my fellowship project is the optimal utilization of computational resources for NWChem, a computational chemistry software package. And the mentor for the project is Dr. Hong Liu from TAG. So there are several objectives and goals for this project. And as we all know, the modern architectures for the physical computers have been evolving and rapidly, and especially with increasing degrees of parallelism. And several of the features of the hardware includes parallelism. There are fast interconnects and communications between hardware, and more memory, and in terms of bandwidth as well as access speed. And particularly there's a complexity of hierarchy of memory accesses. And then there are added accelerators for specific types of applications and for more general purposes like GPUs. And then there are more powerful data storage. So these factors combined with the software itself and how the software will be able to take advantage of these different hardware features will define how the algorithms will perform, the particular applications will perform. So the performance of applications will depend on the library, of course, and whether the library used are optimal for the underlying architecture. And then from a application standpoint of view, how to configure the number of nodes, number of cores, and how much amount of memory. And then all these factors will have a very visible impact on the application performances. So it's often a not so easy task for users to make judicious choices to make efficient uses of these resources. And then from a developer point of view, how to analyze the bottlenecks of these hardware features, like whether you are hitting the roof line of the memory or the I/O and so on. These are factors more important than even the CPU speed, which will define the performance characteristics of your code. So analysis of these factors will be very informative and very important for understanding the features of the software itself and how to develop optimal software and how to use this software efficiently. So the goal of this project is to understand these features for a set of calculations, particularly for quantum chemistry calculations. And then there's some change of the objectives during the project. Initially, the plan is to have some in-depth analysis of the data movements and then come up with a roofline model for the application. And then we also want to do now we want to more focus on combining a data set of performance metrics for these type of applications. So these will be very informative for users and perhaps even for data centers to how to acquire optimal resources for the data center, and also for users to make judicious choices of how to use these resources. So the package we have using is NWChem. NWChem is one of the most popular molecular chemistry simulation package. It's been designed to be parallel from the very beginning, and there are large numbers across the country in different centers, especially and some of the large computing centers. It's a comprehensive package. It contains essentially all the capabilities of the modern electronic structure theory, and the workhorse of these packages includes, for example, DFT methods and coupled cluster methods for computational chemistry calculations. So it's also a primary candidate for porting to the next generation of exascale computers, and there are active work of porting NWChem to the next generation of platforms, so there will be a huge benefit for users to understand the features of this software, especially for us who have been supporting this software. It's essential to understand these features and to help users to make informed choices of the resources and so on. So the tool we use for analysis, there are a couple of tools for analysis of the performance of the software package. There are some commercial ones, which is very useful. And these are installed at some of the major computer centers, including TAC, and it's ARM-forged. So there are two components to it. One is a debugger, the other is a map, which is a performance analysis tool. And then there are Intel suite of tools. For example, the Intel suite of tools we have used for this project so far includes the Advisor, MPI, TAC, and Zhiyong. Essentially, these allow you to collect features of the performance in terms of the communication features, MPI communications, memory analysis, and so on. And besides that, there are new packages, for example, from new architecture. AMD has been developing their own suite of tools for compilation, debugging, and performance analysis, which is AMD ROC-M. The new features of the Intel and AMD tools is that they try to integrate the let's say CPU and GPU analysis and so on. So one of the plans is also to extend the use the analysis to using the different tools from AMD. So that's the analysis tool. The hardware we have been using so far is mostly from the Stampede architecture. That's the Intel architecture with dual sockets. There are 48 cores on each target, and there's a relatively large memory, 192 gigabytes of memory, and there are a hierarchy of storage and cache available. So what we have been focused on is a relatively small set of calculations and not too big calculations, just to, for example, to get a initial data set and figure out how to aggregate data. So we have been using very large number of cores and very large number of nodes and so on. So these are mostly designed to just to have a data set which should have all the essential metrics we need, but we haven't been spending too much effort to understand how these will be scaling with a large number of nodes, and that requires calculation of larger sizes. So we basically use the two nodes and use the 48 cores on each for each node. So the set of calculations we choose to use is the quality assurance suite of calculations from NWChem itself. There are about 600 calculations there, and there are about 200 quantum mechanics calculations. So our analysis will be focused on the quantum mechanics calculations. So we develop a script which can run these calculations automatically and then generate the hardware metrics during the performances. So basically, we will use APS from the Intel tools, and we collect the standard set of metrics, including elapsed time, MPI, memory, and how much memory is used, how much disk I/O is used, and particularly how the vector capacity of the packages are being used. So these are standard subsets of the metrics, and you can also get more finer grain metrics for each of these categories. And then, of course, there's a VTO analysis for the roofline model, and hopefully we'll be able to add more to that later. So this is a particular result. This is one of the largest molecules we have used. This is beyond the 200 or so quality assurance data set we have used. It's one of the calculations I used some time ago, and that's relatively large. There are about 100 atoms, and that's a more meaningful calculation in terms of the real application performances. So we use the MPI to understand MPI tool performance collection tools to understand how this type of code will be able to utilize MPI communication metrics. And basically, this is used as a more in-depth analysis situation to understand a overall picture of the metrics. So for this particular calculation, and it's very typical of the some of the quantum chemistry calculations. This is DFT based, and there's a generation of integrals and storage of integrals. So there's quite a bit of disk I/O involved in the calculation, and then some of the libraries can make heavy use of the vectorization pipelines and so on. but the majority of calculations may not be able to use to take advantage of the vectorization capabilities. And it also very much depends on how you compile the code. And so what we have found for this one is there are the very low floating point utilizations. And so there are quite a bit of disk and memory stalls. So and so the point here is try to collect these matrices, and so user will be aware from a user standpoint of view. User will be aware of whether the code can make use of memory efficiently, and if there are ways of allocating the memory more efficiently, and then you can see, for example, whether this vectorization vectorization utilization is optimal for the calculations, and so on. For this particular calculation, it's interesting that in the compilation, we try to compile the code so that it's going to use a mixed vector AVEX, so 256 and 512. So there is a switch between the context, and since the underlying vector set itself is 512, but it's down loaded to 256 for efficiency purposes, and we are still trying to analyze in more detail how this will impact the code itself, and hopefully we can include that as a metric, so to inform users how to choose the optimal vector instruction set for the compilation and so on. So that's basically the procedure we have been following: run the calculations, collect metrics, and for specific calculations, then analyze the data in more detail. And so in summary, so we have analyzed and collected the performance metrics for a relatively large set of initial quantum chemistries, but these are typically small calculations. And then in the future, we'll try to scale it up to include more realistic calculations, like the one I discussed for the calculation I used before. And then for currently, this is still for quantum chemistry only, and then there are a large number of other different domains of high performance computing where people have heavy calculations, and a collection of these characteristics for different applications in different domains will be quite interesting. Okay, finally, I would like to thank the ECSS fellowship program for making this project a possibility, and especially I would like to thank the fellowship program staff, Bob, Jay, and my mentor, Dr. Hong Liu. And I thank the fellowship fellows, and it's very informative program. We have learned from each other quite a bit. And finally, thanks for my managers, Ruth and Randy, and the other SRCC colleagues, which make it possible for me to participate in this program. Thank you. Thank you very much.
Great. Thank you, Zhiyong. We're a little over, so we just have time for maybe one question. Do we have any questions or any comments?
I'll stop share.
Okay. So, Zhiyong, I actually have a couple of questions, but I can follow up with you offline regarding bit building and benchmarking on AMD, and also the impact of the AVX512.
Yeah. Great. Yes. Great.
Let me just since we're a little over, let me get on to Sinclair's presentation. So Zhiyong, you could go ahead and stop sharing.
Did I okay, sorry. Now it stopped.
All right, great, thank you. And Sinclaire, good to see you. Thank you could make it after you. I guess you were keeping an eye on a take-home exam. So go ahead and you can start sharing and begin your presentation whenever you're ready.
Okay, one moment, please. Actually, so right now I can't seem to share my screen. I apologize. Let's see.
I want to make sure that you have permissions to share.
I might have to come back into. If you can wait 1 minute, I might have to come back into the
Okay.
presentation. So. Hi, everyone. Can you see my screen now?
Yes, now we can.
Okay, I'm so sorry. So let me get started. Yes, so I'm Sinclair Im from Yale, and I'm a graduate student with an applied math machine learning background. And this project was a great opportunity for me to learn more about one of the really hot domains in machine learning, which is material science. As far as machine learning goes, material science is a relatively unexploited domain, but it's quickly becoming exploited. But there are some issues with using machine learning and material science, and we'll talk about that in a little bit. But most of it comes down to just having bigger and better data sets. So I guess big picture is this project was to estimate substitutional defect formation energies in Maxines. And so traditionally, the way that people go about this is to use DFT, or first principle DFT calculations. And as we've seen from just the previous presentation, DFT takes too long. And if you're trying to synthesize materials, overall development turnaround time is very time consuming. So one of the goals here was to develop machine learning models to predict formation energy with reasonable accuracy, to identify features that were pertinent to different classes of materials, and to speed up the overall design process of materials. So beyond coming up with some way to screen for possible materials that might be useful to synthesize either computational or experimentally, maybe there would be a way to come up with some good design principles if you came up with a very nice sparse feature set. So as I mentioned, DFT is just can be very time consuming if you're trying to iterate over all these materials to come up with a nice smaller set to maybe synthesize. But in this case, Maxines are a class of two-dimensional inorganic compounds, which are relatively recent in the last decade. And I'd say there's a lot of research about these materials. They have a lot of promise for batteries, supercapacitors, and composites. Overall, there's been a lot of research because machine learning holds the promise to speed up the overall research cycle. And I think we're seeing that. If you look in the literature, like every month, there's a number of papers either published in peer-reviewed journals or just online. So this is a very exciting area. There's a lot of recent research, as I just mentioned, and here's two. I could point to so many, but here's two that relate to this kind of process for either Maxines or other materials. In this case, it was a fairly small data set that we're working with, just 114 observations for defects relating to titanium nitride and titanium carbide. The models that we used and the resources, initially we weren't using some of these off-the-shelf tools. We were kind of rolling our own tools, but then as we try to develop like more robust and speedier pipelines, we moved to off-the-shelf material. So scikit-learn, the materials project, and MapMiner. And these are all like extremely useful tools, and we're trying to push some of the extensions that we made back into these libraries. So we tried a lot of different approaches in machine learning. And I guess for this kind of like small case study that I'm showing, we ended up with a fairly simple approach that was robust, and that's random forest regression, which is a supervised learning algorithm just constructs several decision trees, and then you come up with like some sort of average response for those trees. And it's pretty powerful. It's robust. You don't have to scale, which is really useful in this case. But it's hard to interpret the model sometimes, but that's kind of the trade-off that we had and that you have in a lot of machine learning. So here's some of the preliminary results for this particular case with the random forest regression. It was a fairly good RMSE, but the data set was relatively small. Again, just 114 observations. So while it's promising, we have more work to do. The key features that we found that were most important in this particular random forest regression were fusion heat, metallic radius, ion energy and atomic volume, and electronegativity. We had at some point thousands of features, whether they're composite or structural features, and we found that many of those were just not very influential. So for this particular project, I would say the amount of computational resources used was not extensive. A lot of the initial development work I could do locally on my own box. And then as some of the pipelines got more extensive, I could do that in an on-campus cluster. I think that's something like in the future when these data sets get larger, that we'll find that we'll need substantially more computational resources. But for the time being, it wasn't very extreme. In terms of future work, I think there's room to do like a lot more basic blocking and tackling in this particular set of materials. But overall, I think we're looking for opportunities where we can use existing large data sets, like for example, the Open Catalyst project, or to somehow augment these data sets, because you really see the power of machine learning with much larger data sets. And then I just want to thank my mentor on this project, Dr. Vineet Sharma, the PI and the program, Jay and Bob, the rest of the fellows, and the computational resources of XSEDE and the Yale campus clusters. Thank you.
Thank you, Sinclair. Do we have any questions for Sinclair? Or for any of our speakers?
So what DFT method do you use?
Is that directed at me?
Yeah.
Okay, so we weren't using DFT. I was not using DFT. I took the output as DFT as a given. So the idea behind our project was to take in all these estimated DFT calculations as our inputs.
Okay, thank you.
Mm.